This is Dr. Perkins, and in this video we're going to talk about enzymes. So what is an enzyme? An enzyme is a particular kind of molecule that acts as a biological catalyst. So what exactly does that mean? Biological means that an enzyme is found in living systems such as mushrooms, humans, or even plants, for example. And a catalyst is something that speeds up the rate of a chemical reaction without being used up in the reaction. But what is a chemical reaction? In a chemical reaction, molecules such as this amino acid are changed into something else. For example, they could be assembled into organisms like cells, or cells can be assembled into tissues or even organs such as this heart, the ear, or the nose. Or there are sometimes toxic molecules produced that the body needs to get rid of. And so our bodies change those molecules into something less toxic. For example, one toxic compound that builds up in body cells is hydrogen peroxide. And our bodies convert it to water and oxygen. You have two molecules of hydrogen peroxide, give you two waters and an oxygen molecule using the enzyme catalase. And when one molecule is converted into another, this is called a chemical reaction. All chemical reactions catalyzed by enzymes would occur naturally on their own without enzymes, but that could take years, and in living systems, products are needed immediately. Otherwise, the cell could die. And so enzymes actually function to speed up the rate of a chemical reaction that otherwise might take an extremely long period of time. And let's look at how that happens. Enzymes speed up the rate of chemical reactions by lowering the amount of energy required to start a reaction. To illustrate this concept, I'm drawing a graph. On the y-axis, you have the amount of energy required to start a reaction, and on the x-axis, you have the progress of a reaction, or the reaction path. In this reaction, reactant X is converted into product Y. But for X to become Y, we need an input of energy shown by the black line. In the presence of an enzyme, however, the red line shows that X does not need as much energy to become Y. The amount of energy required to start a reaction is called the activation energy. The activation energy without the enzyme is much greater than it is with the enzyme. Because of the reduced amount of energy needed, enzymes speed up the rate of the chemical reaction. Let's talk about how an enzyme works. Let's start by drawing a picture of an enzyme. An enzyme, usually a protein, has a little pocket in it called the active site, and this is where the reactant fits. In an enzymatic reaction, the reactant is called a substrate. This enzyme could be catalase. Hydrogen peroxide, a single molecule, fits into the active site, and an intermediate called the enzyme-substrate complex is formed. And the chemical reaction then takes place in the active site of the enzyme. And then an enzyme product is formed. And then the two products, water and oxygen, are released from the enzyme. For any enzymatic reaction to occur, three properties always apply. One, the enzyme is unchanged by the reaction. Number two, the enzyme can be reused and can catalyze another reaction. And number three, enzymes are kind of like a lock and key. An enzyme and its substrate bind together like a lock and key. Because of the unique shape of each active site, only one substrate fits into the active site, and only one chemical reaction can be catalyzed by one enzyme. So an enzyme and its substrate have a unique relationship. The enzyme is the lock, and its substrate is the key. For example, the enzyme catalase specifically recognizes hydrogen peroxide and will not bind to any other substrate. But let's talk about other characteristics of enzymes. All enzymes, for them to function at their best, need what are called optima. They need a best pH and temperature. For example, the enzyme disaccharidase digests a disaccharide, which is two sugars linked together, into two monosaccharides. This enzyme is found in the intestine, and therefore its optima are neutral pH and body temperature, 37 degrees Celsius. In fact, an enzyme's optima normally reflect where the enzyme is found in nature. As another example, the enzyme pepsin, which digests protein, is found in the acid stomach, where there's a lot of hydrochloric acid. This graph shows pepsin enzyme activity at different pH values. And you can see that the activity of pepsin peaks at about 2.0, which is just about the pH of the stomach due to the presence of stomach acid. What temperature do you think pepsin functions best at? If you said body temperature, you would be correct because pepsin is found in the human body, which is 37 degrees Celsius. Enzymes that are placed in extreme conditions lose their shape altogether and they stop functioning. You can see that pepsin is not active at pH 0 or pH 4. But if you put an enzyme into boiling water, let's look at what happens. We're all familiar with frying an egg. Much of the protein making up the egg white is called egg white lysozyme, which is an enzyme that digests polysaccharides. It's not really white till the egg is cooked. This is a good example of how, with the naked eye, we can see that an enzyme undergoes a complete change in consistency and appearance through exposure to extreme heat. This is a phenomenon called denaturation. The enzyme is now completely inactive. It goes from the natural state to the denatured state. And I hope you enjoyed learning about enzymes.